All right. So related to something that David, you actually brought up in the Discord, like a question you had uh, regarding. So what did you say exactly? Let me see. Because it was a really good question, and we kind of talked about it, but it just got me thinking about it being a good topic for stream. Yeah, today you read an article about an HIV patient having been cured from the illness due to a bone marrow transplant from a person with certain genetic mutation, which is in CCR5, which is kind of famous for having. Actually, why are we looking at this? CCR5. For having a mutation, I believe I have it somewhere because I don't have it memorized. The mutation is a Delta 32 variant, which, if I'm remembering my human gene nomenclature, means that there's a deletion at the 32nd amino acid, maybe? I should probably look that up. I don't know that for sure. CCR5 Delta 32. What is it specifically? Let's see. Oh, a 32 base pair deletion. I'm sorry, that's wrong. 32 base pair deletion within CCR5, which, by the way, is CC, chemokine receptor 5, widely expressed in several immune cells. We're looking at the gene right here. Cyanophyte, welcome. Haven't seen you in a while. What's up? Sorry, I'm reaching over to get a drink. I'm going to start with a smooth IPA and finish off with water. All right. Cyanophyte, you might be interested. I forget who it was specifically. In the future, possibly this Sunday, if I can get my act together, I was going to try and 3D print a little tiny maze that maybe we can drop some tardigrades or nematodes into and see whether or not they can navigate through that maze. I don't actually know if my 3D printer will be able to print something quite with small enough channels that it won't end up being proportionally like just a giant labyrinth for the poor things. All right. So this is kind of a terrible view. But CCR5 is this blue thing right here. Let's zoom in a little bit. Zoom in. So I'm just wondering where this 32 base pair deletion is. And the reason why I wanted to look at where it was before we start talking about genome or genetic engineering, I have a little bit of my own genetic data, and I was going to see if maybe it happened to contain any information on whether or not I carry this variant, which would be fun, but also a surprise because it's fairly rare, I think. It's been a minute. You took a break from streaming to touch some summer grass. That's fair. Yeah, I'm glad you got outside. Yeah, I hope the maze works well too. I mean, we'll see. I know relatively that that can make something quite thin, but going between something I can hold in my hand and something I'm seeing under the microscope, I just don't have the sense of scale there, the change in scale, I should say. All right. So CCR5, people who have the 32 base pair deletion have decreased susceptibility to HIV. Specifically, people who have carried two deletions, one from their mom, one from their dad. Therefore, they have no normal CCR5. I don't know if they have any CCR5. I'm sure if I read through this, it would say. So I'm just looking at this sort of information sheet, just looking at the HIV susceptibility. And so why I'm talking about this, generally it's because there's this idea that David, you sort of suggested and was already sort of in the works, of this idea of, well, we have this variant that we think will cure or decrease susceptibility to HIV. What if we just stuck it into people who have HIV? Examining one's own genetic code on stream is another level of doxing oneself. Well, you know, anything for views. Not really, but you know, it's cool. So let me see. How can I actually show this? So I got to download something quick or open it with Google Docs. Let's do that. Yeah, so let's just start off before we go into, like, you know, CRISPR editing. And then eventually I do want to look at this this trailer for Rampage, a 2018 movie I did not watch that specifically mentions CRISPR in the title. And then I found some other examples of, and I think they're pretty well known, you know, genetic engineering in sci-fi, that sort of thing. Manogard, I definitely want to talk about Stargate, because they got it. They didn't get it right, but they didn't get it wrong, and they got it right. They almost got it right. They were so close to getting it right, and they talked about it so naturally that I was kind of surprised in retrospect to think, like, that was like 2005. Like, yeah, scientists kind of knew that maybe we'd do this someday, but I'm surprised it made it to TV. All right. Google does not like that document. I need to download it really quick. Scanning for viruses. It's scanning my DNA for viruses. Okay, here we go. I do not think this contains other doxing information. So this is from an Ancestry DNA test that I took a long time ago at this point. So Ancestry DNA and other of these types of tests, what they do is they examine individual variants within your DNA. So it's very, very targeted. It's called a single nucleotide polymorphism test, or SNP, or SNP, array genetic test. It's kind of like the old style. I'm just looking up. So a SNP is are these places in the genome. So we are largely the same as everyone else. Like, on average, there's like no difference between any one particular human or any two particular people, humans, but there are specific differences, and those differences sort of add up and make the uniqueness that we have. So a sort of word for those specific differences are a single nucleotide, because it's like one nucleotide polymorphisms, right? So that means, like, polymorphism just means many types. So person 1 at this position has an A, person 2 has a G, and person 3 has an A. Easy enough. And then you have CAA. So see, 1 and 3 were the same. Now 2 and 3 are the same. Now 1 and 3. Now 1 and 3 again, right? So you can test individually each one of these. And that's what most genetic testing services, that's what most did. I think we're getting to the point where some are actually offering full genome sequencing. But basically what we're doing is, like, each SNP has a name, or RSID. I forget what the RS stands for. It's just a SNP identifier. It tells you that then we know where the chromosome, we know the position, and then we get our 2 alleles. So you have 2 versions of every single gene, and that is because every single person is created from a sperm and an egg, each of which would had 1. Bring them together, you have your 2. So, for example, at RS4477212, I have a T and a T. Great. We can actually look this up. Let's see. SNP database. I forget exactly where that is. This is not quite what I was looking for. All right, so this one And to be honest, I'm not usually quite sure how to read these, like this notation. The alleles are A and G. So I have two T's. So I don't know if that's very rare, or since A pairs with T, what they really meant is sort of the opposite. It depends on the call, I think. I don't know. I don't ever think about these types of data. But this is not what I was thinking. There's like SNPpedia, SNPedia. Here we go. I feel like there's a little bit more information here. So we know things. We know things about certain ones. So if I click on this, oh, so there isn't a page on this. So at least for SNPedia, it hasn't risen to the level of any amount of interest. That was just a random variant, but there's tons of other ones. You can't help doing an automatic word search scan for Gattaca. Yeah, Gattaca is another one of those. Gattaca's good too because they did genetic engineering without really getting into the details. They were just like, yeah, this is a thing, we can do it now. Here's the consequences. So that was fun. I'm sure Gattaca shows up. Oh, Gattaca has to show up in the human genome thousands, maybe millions of times. Here, we can look right now. Let's see. So if we do tools, blat, Gattaca. Oh, it wants 20. Let's do Gattaca. Actually, we'll need more than that. I'll just put Gattaca. I'll just put 4 times. Hey, look at that. Gattaca. Right there. So apparently in the human genome at, where are we? Chromosome 4, we have the word Gattaca. It says it oh, hey, Alt Miss Frizzle. Welcome. Yes, it did force me. Probably because it knew that if we did less than 20, it would just show up way, way, way too much. This isn't even a gene. This is just some random part of the genome. Whatever. How are you doing, Alt Miss Frizzle? Hope you're doing well. We're just looking at my DNA. So anyways, like any normal human, I have lots of DNA. I have lots of variants. So I'm just scrolling. Lots and lots of variants. So the question is whether or not I have... Oh, this is the other thing. Each test doesn't necessarily test for the same SNPs. So there's a very, very good chance that even though I have all this information about my own DNA, I might not have the information I need to look at CCR5. So, I had this somewhere. I had the CCR5. Where are you? Where are you? You think it's fascinating that a couple decades ago we knew next to nothing about DNA. Nowadays we can look it up and probably read it like some kind of language. That's not too far off. Yeah. Where'd it go? Shoot. David, I believe you joined the stream after we chatted with James Gurney and David Hewlett about the book The Genesis Machine. But that book is a very good sort of record of the last like 10 years ago minus another 10 to 20 years ago, kind of that period of time that you're talking about where we went from like, okay, we know what DNA is, we know it's there, we know it's a code, but also we don't know that much about it, to suddenly we can just sequence it like that. Very good if you want to look that up. All right, I thought somewhere, sorry I'm flipping through all these. Let's go back to CCR5. Ooh, if I don't have the mutation, okay, I don't want to do a poll, straw poll. Would it be interesting to actually go through what is required to like CRISPR the CCR5 deletion into my DNA? That's something I could do. Okay, but where is the CCR5 SNP? Ah, here it is. Okay. All right. So, oh, it's from SNPpedia. Duh. Oh, hello, Brian. You sent me a paper on Discord to show how the deletion works. Oh, perfect. That's great. Where are you? There you are. Ooh. Oh, this is super informative. Yeah, let's look at this quick. Thank you for that. All right, I'm getting some yeses to that. All right. Well, I hope we'll do that. But I hope it's interesting. But honestly, some of it's so trivial that it's like just kind of like moving some letters around. But we'll do it nonetheless. Just with a huge asterisk of, don't try this at home. All right. So let's see if we can go away. All right, so this is the HIV infection process. So these are HIV viruses. So they attach via CCR5, which is this brown squiggle. It's called a transmembrane, or it has transmembrane domain. So that means part of the protein is outside the cell, parts inside the cell, and parts halfway through. It looks like we have CD4, this other protein coming in. They kind of interact. I don't know which comes. Oh, actually, I guess CD4 comes first. And, oh, I see their arrows now. That's sort of counterintuitive, the direction it's going, but whatever. So that allows for membrane fusion, and then you get your HIV infection in the cell. So that's not good. I don't know. Ah, here we go. Well, that's easy. So this is how it works. See, this is the fun thing about molecular biology, is it really comes down to just pieces fitting together sometimes or not. So in a normal wild-type human who has CCR5, this co-receptor, which has many other functions and is probably very necessary. In fact, I'm pretty sure people with this mutation on the right-hand side are at really high risk for aneurysms. So they have it, they can get infected. This other one with the deletion makes it truncated protein, it looks like. So protein that just isn't as long as necessary. It doesn't have a function, and therefore you're resistant because it doesn't have both of the two-part component that's required for the virus to, fuse with your cell membranes. Structure equals function. Absolutely. Alt Miss Frizzle. They're just mad they took away teaching structure of biomolecules for your standard, but that's the whole point. That is the point. That's ridiculous. Second figure says it introduces a premature termination codon. Oh, here it is. Well, here's the protein. These are some really pretty figures. It's very detailed. They didn't really have to do all these, but still, that's cool. All right, figure 2. The CCR5 delta 32 deletion site is denoted with a triangle found just before the 2D7 binding site. Mutation at the site results in a premature stop codon. Thus, the deletion of all protein structures after this location, resulting in the loss of the 2D7 binding site and cytosolic CCR5. So, right here. Where's the, let's see, N to C? Oh, yeah. So this deletion right here removes everything afterwards, or at least partially everything afterwards. All right, you're only required to teach function. Okay. You're still teaching structures to your honors kids. Good. And you decided to teach your level like honors 2. Oh, great. So you're still teaching it. Nice. Sounds like a stupid idea. What are you supposed to say when asked why a certain protein does certain things? Magic? I guess unfortunately in Alt Miss Frizzle's state, city, school district, whatever, I guess you're supposed to. Unless you're in her specific class, then it sounds like you're getting a good, wide-encompassing, proper education in biology. Okay, that's interesting. What is this paper? I didn't even read the title. Oh, wow, it's like the topic. Biology, gene editing, and warnings of the future. Oh, wait, I opened this. I already had this. Yeah, I just didn't scroll down. Cool. Texas. We know of Texas. It's known to us. All right. Ooh, cool. There's some advantages and disadvantages. Thank you, Brian. This is like perfect. Let's see. If you happen to have the link, would you mind putting this in the Discord when you get a chance? In the the primary literature thing. Otherwise, I will afterwards. I have the link somewhere. All right. Anyways, CCR5, the chemokine receptor protein. It has a couple known SNPs, so single nucleotide polymorphisms. Let's see. What chromosome is it on, though? It is on chromosome 3, and it's at about the 46 million position. So I'm just laughing at Alt Miss Frizzle quoting myself back at me. Okay, 3. Let's see, 1, 2, 3, 4, 5, 6. Oh, why is the human genome so big? It's unnecessary. I bet there's stuff that we could get rid of. 46. 46, 46, 46. All right, we're in the 46s. 46. I got to put these right next to each other. 464. See, I don't know if my test will even cover this area, so we're going to have to see. Oh. Hold on now. Marcus as a biohacker arc. Yeah, I am become biohacker. Excuse me. First of all, this. No. This has to be. Wait, what? Okay, I have read 00 for this. I don't think this is in the correct spot, but that's very close to the gene. That means I'm missing. Huh. So I don't, when I prepare for stream, I pull up some things, I read some things, but when I want to do something actively, I don't do that. So things like this are a surprise. I'm going to have to look this up. What exactly am I missing here? And is that in the CCR5 gene? I don't think so, because the position is a little bit off. Yeah, learning some hard truths on stream. How much of the human genome can we get rid of safely? Imagine proposing that to an ethics board. You would be met with such derision. Okay. Hold on, I gotta look up that particular SNP. Nothing. Okay. We gotta look up that specific location. Am I on the right chromosome? Okay, chromosome 3 and this position. Okay, we're at chromosome 3. I think we can just do that. Maybe it won't like that. It did not like that. Let's do chromosome 3. So this is, oh, I didn't even introduce this. This is the human genome browser, or a human genome browser. It's one of many genome browsers for many species. Yeah, sorry, I didn't talk about this at all. And it's probably super confusing. I'm just looking at these blue ones, which are telling us what the genes are. There's conservation scores down here, common SNPs down here, and a bunch of other random stuff. In fact, I might as well hide some of these things because I don't know what they are. We'll keep allelic variant phenotypes. That might be interesting. Okay, let's go here. All right, so this is a place of a common SNP, which is why it showed up in my test. Zoom out. But where are we? Zoom out. Zoom out. Zoom out a lot. Okay, we're not. Wait. Too much. Does my institution pay for this? No, absolutely not. It's completely free. Here. If you want to go poke around on a genome of a, any number of model organisms, including humans, you can just go there. You can go down to the DNA level, like we just saw, but it sort of disappears as you zoom out just because there's too much. If you go to Tools and BLAT, that's where you can do what I did before, where you can search it for a specific sequence. Can you compare between organisms? Not yes and no. The easiest way to do that is on the human one. There are these are tracks. The ones that I'm highlighting right here are showing the conservation between other organisms. But you can certainly hop between different organisms' full genomes and kind of look around. All right. Full? Full. All right, I'm just a little confused about what I just found. Why? Where'd CCR5 go? I've lost CCR5. Oh, it's way over here. Okay. So first of all, looks like I have a deletion somewhere, but it's not in CCR5. It doesn't seem that there's many that in my particular test, which is a shame. I don't think it covers CCR5. Or maybe it does, and I just didn't scroll. Let's see. The smallest, okay, 3, 7. No, it doesn't look like it. So it looks like I skip all the CCR5 stuff, unfortunately. But that's okay. Yeah, if you use this in class and ever have any questions, let me or just someone in Discord know. Lots of people use it for various things. Let's see. Okay, well that was sort of exciting, but ultimately nothing. Wait, am I blind? No. So between here and here, unfortunately, there's a gap between the SNPs. There's no 3, 7. Shoot. Oh well. All right. Well, let's do some genome editing. So I guess the point though is that we are, the point of the stream, at least for me, is we're getting closer to this point where films like Gattaca, which I saw is this website calls the scariest sci-fi movie on Netflix. So basically the premise of Gattaca for those who don't know or haven't seen it, is there's some future where there is a eugenics society, so parents are widely given the option to genetically alter their children, but although they do this with the best intentions, what ends up happening is it stratifies society where everyone is constantly checked for their DNA, and people only with certain traits are allowed to carry certain jobs, and those jobs, unsurprisingly, are very much tied to class. So it creates a higher and a lower class stratification based purely on genetics, but those genetics have been altered by, prior to birth. Making myself bigger. And the follower is bigger. I had to up my follower goal. I didn't really celebrate, but it reached 100 about 2 weeks ago. All right. So what do we need? Because sort of like the thing is, this is the idea, because we're getting closer and closer to this. That was science fiction. Miss Rizzo, you have to watch this. It's good. If you have, I think there's a little bit of nudity in it, not that bad. But if you have upper level high school students, I remember, after having my parents sign a permission form, I remember having like a screening of Gattaca in my advanced biology course. Hello, Megan. Yeah, Gattaca is really good. You watched it for the first time in high school. I think I had seen it before then, but then I saw it in class, and we had a discussion about it. And this was pre-CRISPR, right? So that was sort of even before the mainstream awareness of genetic engineering. I think I've already said this, but right, the point of this is that in CCR5, or honestly any number of genes, if we find out that there's sort of positive components to mutations of those genes, maybe we can delete. Oh my gosh, that scared me. But thank you, Lil Dare, for the follow. Completely lost my train of thought. CRISPR, gene editing. Yeah, so the idea is if we find any number of genes that have positive effects when they are mutated a certain way, or maybe have a certain SNP, like these here, maybe we can alter them in people who might need protection. This person had acute lymphocytic leukemia, so they required a transplant of stem cells. And I believe they also had HIV. And this was sort of a clinical trial to take this chance from this person who really needed a stem cell transplant, take it as a chance to possibly also cure their HIV. So when we call something like in biology, so like something not dead, something that lacks a gene, we say null. So CCR5 null blood cells, these are blood cells that do not have CCR5. They're resistant to HIV entry. So they transplanted CRISPR-edited CCR5 ablated, meaning it was knocked out, taken out with CRISPR, hematopoietic stem cells and progenitor cells into a patient with HIV infection. So that's what the CCR5 deletion was for. But also they have the acute lymphoblastic leukemia. That's just what the stem cells are for. Let's see. So their leukemia went into remission with full donor chimerism. As far as I know, what that means is the patient no longer had blood stem cells of their own, only the donor, which is what you want, because their own stem cells were their own cells were the ones that had the cancer. And then the CCR5 deletion persisted for more than 19 months. Probably continued after that. They just didn't follow past then because they wanted to publish. and they didn't have gene editing related adverse defects. Let's see. And then did they... Okay. But their problem is here that they only really had a deletion of about 5%. In 5% of the cells actually had the deletion, which is good, but CRISPR is not perfect. And clearly they weren't able to get everything in the stem cell population. All right. So let's say we want to do this in my cells. Now, am I going to be able to actually use the application I want to use? Because it does require a subscription. Oh. SnapGene. All right. So let's grab out... I actually don't work in humans, so I don't know where the best place to get it is. CCR5 human sequence. We'll figure this out. Does anyone in chat know where the worm-based equivalent? Oh.
Howdy.
Hey, Defested. Thank you for the resubscription. Howdy to you too. You don't happen to know where we keep the all human gene sequences, do you? Is it just UniProt? Like, what's the worm base, the fly base equivalent, but for humans? Is there human base? If we can't get the sequence, then in the nucleus. Thank you. I didn't think to look there. Ensemble, UniProt. Okay. I'm here. Sequence. Here we go. I don't want the protein sequence. I want the DNA sequence though. NCBI. Oh, great. Great. As usual, you're going to be doing some housework while listening. Well, thanks for stopping by regardless. All right, so we have the human sequence here. I don't know if I'll actually be able to use this. So this is CCR5. Oh, thank you. It detected a common feature. Add one feature. And here's our gene. We are missing all the codons. Don't get arrested like the one guy for CRISPRing this gene into embryos. Well, I'm not going to. This is all hypothetical. This is definitely not a how-to in any way, shape, or form. Let's see. Okay, this is great, but I actually do need the codons. Where are the codons? Yeah, I'm trying to get the transcript. I just need to see where the codons are, because I think a lot of this is just non-coding sequence. Gene table? Transcript table. Okay. Show me. Download. All right, so it makes a bunch of the same protein. Ugh. I guess I'm downloading that. Oh, cool. Not descriptive at all. Well, here, we had this nice picture right here. I can just do the the dumb way to do this, which is simply to find this site that we want to delete by searching the sequence for that protein. So if I do Control F, we're going to find protein sequence. We're going to find SQYQFWKN. Yeah. Fine. Show me the translations. View. How do I translate this? Nope. 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 Also, sorry if that was really loud. There's a variant RNA. Oh, thank you. I will look at that as soon as I figure out how to translate this. On all mine, it usually is automatically translated. Also, how dare it not just find whatever I want immediately? Little yellow and green air. Oh. It doesn't detect. Oh. Here we go. So thank you for that. I did not know what button to press. So what it wasn't showing before was open reading frames, which are kind of like the suggested, like where a protein might be. I think this came up before as a question in another stream of like, how can we tell, where a gene is? Well, if we click on it and look at it, all genes start with an ATG. So all this really did was go through and like look for any ATGs. It had to have missed some. I think there's some other things that it looks for, like it has to be of a certain length. Yeah, actually, so ATG, GAG, and then a TAA almost immediately. So it knew this wasn't a protein because it stops almost immediately. Okay, let's try that again. Here we go. So that's the protein that we want to delete. Let me just go ahead and go back to our genome browser, wherever that was, because I do want to see. Okay. So I didn't look at the structure of the gene at all. Does it have an intron? No, it doesn't. Oh, perfect. Perfect. So it doesn't have an intron. So that means it doesn't have an extra sequence that doesn't show up in the protein. So that means this orange bit right here is just the protein. That's fantastic. So I'm just going to select from here to here. Ends at a stop codon. I'm going to add a feature. We're going to call this CCR5. It's translated CDS. What color should it be? It should be orange. And I'll turn those off. Fantastic. There we go. There's our CCR5. This is going to be our deleted thing. Deleted. This is the tricky bit, though. I need to figure out what the actual deletion is that gives HIV resistance. That's another step. We'll just call this MISC feature. All right, so that's what's probably deleted, or at least part of it. It said like the deletion was like here, about. So let's go back to this paper. Okay, I don't need these anymore. That's just Gattaca. All right, so it's called Delta 32. So let's go back up to like maybe like the first mention of it. That's the, all right. It's found in 11% of Northern Europeans. Interesting. So 11% chance that still I have at least one, since I'm primarily Northern European. But what is this exactly? "32 base pair deletion just before the 2D7 structural loop." Okay. They have some things right here. All right, I might have to go to this other paper, which won't let me. Shoot. All right. 32 base pair deletion just before this SQYQF, whatever. So let's actually just see what happens when we do that. So I should probably rename this. What do they call it? 2D7 binding site. So this is actually the 2D7 binding site. And then a 32 base pair deletion. All right, so I'm just going to delete this really quick and see what happens. I can't delete bases. Fantastic. We'll open a new file then and force it to do that. So we'll take everything after that. I'm just copying and pasting these sequences. And then 32 bases. I take everything before it. So this should mimic our deletion, since SnapGene won't let me do it. So CCR5, whoops, sorry. CCR5 D32. Yeah, add that. All right, so now this is our deletion. If I control A, I'll add a feature, CCR5 D32. Oh, I see you show, I see your, give me another paper. Let me open that. Figure 2. Oh, perfect. Well, there we go. Well, what did I delete just now? Let's see. So GTC, AGT, GTC, AGT, oh, I missed it. What was it? GTC, AGT. Oh, it was starting there. That other paper is written very confusingly then. And it goes all the way until GACA. So that is actually our deletion. Thank you very much. All right. So see, this is why I said it might be kind of boring to watch, because a lot of it is just like, oh, where's that DNA base? All right. So you know what? That other sequence that we opened up, we don't want anymore. Don't save. But this one, we want it. All right, so now CCR5 D32. Thank you for that, Brian. That was very helpful. I don't know what it is, but it's very hard to Google things effectively on stream. So thank you for doing it for me. D32. All right, this is a CDS. And we're going to make this one light pink. So let's check out the sequence. All right, so we're okay. We start at ATG. And there's some premature stop codons. Yeah, that's about, what, halfway through? So we had this sequence right before. So we're going to find that and kind of get our bearings. Yeah. So we have a deletion right here, and then only 30 amino acids down the line, we get a premature stop. So I don't know if it's clear to everyone who isn't biology, like why that happens. But as you can see, when I sort of hover over these are amino acids. Amino acids are encoded by 3 bases, the codons. And each codon, each codon codes for one thing, but then each amino acid is encoded by anywhere from 3 to 4 to 1. And then you have the special codons, which are the stop codons. It is genetic grammar that basically tells some of the cellular machinery, stop making a protein. Not biology-y. Yeah. Which actually reminds me to check those fools at Twitch. They're keeping my emotes still locked behind pending. I don't know why that reminded me, but I thought I'd check quick. Anyways. Okay. So, why is my chat so bad? Okay. They don't want you to have tardigrades. Clearly not. They hate it. But if you go to Discord, we do have them on Discord. I think, Manogard, you must have Discord Nitro or whatever it's called, because I saw you use one in David's Discord. Or the Tech Bandits Discord. Okay. So this is where I get into trouble, though, with this scheme to do this on stream. So this software is fantastic, but it does require a subscription, and I don't know if it'll let me do what I need to do without VPN-ing into my university. But I don't want to VPN while streaming because I don't know what that will do. Also, I'm not supposed to do things on that particular VPN unless it's just for using this program. So I'm not going to do that. So we'll see how far we get. Someone else used it. Oh, that's, Second Drink used it. If it's downloaded to your computer, you should be good. At least that's how your copy is. That you have through your university. So I need to be at my university's network to be able to use most of the functions. Okay, but I'm getting away from this regardless. We'll just see what happens. So I want to delete this bit, right? Okay, first things first, I need to find a site to cut through CRISPR. So this is the sort of wonderful thing about CRISPR, is that all you have to do is find one of the special sites, and I see one right about here that ends in a certain sequence, an NGG, or any base followed by a GG. I don't know if this is a good one, but this might be one right here in the center of the deletion that we want. Check the Discord when I get a chance. Oh, okay, Brian, I guess I can take you up on that offer. I don't know how to activate. Hold on, everybody. I'm doing something quick. Gotta move that off. Yeah, I'll take you up on that if it's not too difficult. I don't actually know how to do that. We're getting myself access to this program. Legal access. Which sounds suspicious. Actually legal access. I just need to move these over and save. So while that's happening, I might as well just pull up and explain whether or not, because I don't know if everyone knows how CRISPR works. CRISPR, I guess that's what I'm typing in. So CRISPR is a 2-component system. Oh, this is a good picture. So you have this protein, which is that gray blob behind it. You're targeting some DNA, which is the blue. So the first part of the component is the protein itself, this gray blob. It holds on to a piece of RNA. That piece of RNA is variable, and that lets you specifically target different parts of the DNA. Once you do that, once it finds its happy little target, it cuts. And after it cuts, that allows you to potentially make a repair. Oh, okay. We're going to hold off on that for just a second. Then I have seen a great video before on YouTube kind of showing the dynamics of this. I think it's Genetic CRISPR. This was a paper from like 2014 or something like that. CRISPR's gotten way too popular. It's very hard to find specifically. CRISPR Wild West. Come on. I'm looking for a specific video I saw a long time ago where they discovered exactly how the Cas9, this protein, this gray blob, finds and cuts its DNA target. Targeting. Yeah, there's just way too many way too many videos about CRISPR. Won't let me find what I'm looking for. Too many things about Jennifer Doudna. Let's see. Oh, info in Discord. Thank you. Okay, gotta download something really quick. So I probably should close this out. Ooh, it's the new version too. Awesome. Yeah, so please kick me off this once you're done, or once I'm done. Ooh, sorry. That was probably loud. All right, we're reinstalling. Please hold. I see, okay. Yeah, we have a site license, not individual licenses, which is both good and bad. All right, I'm looking for the specific CRISPR yeah, the specific video. It was like a simulation back before we had anything about Cas9. Its shape, its like broad function. It was someone doing a really bad Western accent. Come on. Where are you? Run SnapGene. I am technically a former lab member. Well, thank you. Not only do you subscribe to me, Brian, but you also give me licenses for far too expensive programs. Go away, SnapGene viewer. Gotta agree to these. Ooh, do I have to do do I have to do this part? Oh, optional. Ew. Okay, I'm gonna do that part off screen because it's something else. As long as you don't get in trouble. Let's see. You finished your housework today, mowed the lawn, and installed insulation into your soon-to-be basement bathroom. Oh, nice. That's exciting. I love a bit of housework. Also good on you to, actually like be able to do something like install insulation. Eesh. Oh my gosh. Don't show me a video. All right. Finally. Okay. We're back. Now we can finally do some CRISPRing. Yikes. So. Got to open those recent files. All right. This is already much better. All right, so great. We needed a couple things, right? We need that special RNA. So that special RNA is going to bind at a specific site. It binds NGGs. So if I look for NGG and I search through them, all of these highlighted sites are potential loading, like landing spots for the Cas9 protein, which will then come and cut. So the NGG, for example, is like right here, the bit that actually doesn't sort of connect with the DNA. So we need to design a guide RNA, is what it's called, for probably something just like this. So thankfully, lots of smart people have made tools for this. So I'm going to search for one of those tools. CRISPR. I believe this site still works. All right, so we are going to do the human genome. Oh, I guess I should start at step one. Sequence name, so this is CCR5. And then we will put in a single genomic sequence. It's saying typically an exon. So that genomic sequence is going to be, we want to keep it pretty targeted. So we're just going to highlight this bit because we want to land kind of in the middle right here. So I'm just going to go ahead and copy that. And we'll just paste it right here. So genome, we're doing Homo sapiens. Let's do the most recent Homo sapiens. And then we need to select a protospacer adjacent motif, a PAM. That's that NGG. We're just going to use that. Then wait. So what it's going to do is simulate sort of CRISPR events because we want to cut that one spot. We don't want to cut anywhere else. So what it's doing, oh, it finished already. So what it's doing is looking where I highlighted right here, looking for potential guides. So here's a guide, here's a guide, here's a guide, looking for those and seeing like, okay, but does this sequence show up anywhere else in the human genome? So let's take a look. So they give a couple recommendations. So this is already listed by best to worst down here at the bottom. Up here is the sequence that I gave it. I didn't highlight that, I didn't keep that highlighted. But basically anytime it sees, one of these, it's showing a guide RNA. So this will help us target our mutation, right? Targeting. Let's see. So first things first, there's some scores that we don't care about. It's already ordered it for us. So for our purposes, we don't really care. This is what we care about. We want to know whether or not there are off targets. So whether or not it targets anywhere else in the genome. See you later, Defested. Yes, please go shower and relax. I'll talk to you later. Enjoy your night. So basically it's saying there are 0, 1, 2, 3, 4 targets, off targets, with this many changes to this sequence. So for this top one, for example, there are 0 targets that match perfectly to this suggested guide sequence. There are 0 that are match this guide sequence with a single change to it. I will try not to commit too many genomic taboos. I'm not doing anything no one else has done. Oh, I forgot I had a lurk command. It doesn't do anything except Fossabot says that. Okay, and then et cetera, et cetera. It looks like there are 100 off targets with 4 mismatches. So it's kind of interesting because we can also see, like, so there's one target that has 2 mismatches, and the gene is called CCR2. It's probably related to CCR5, so it has some similar sequence to it. So hopefully that won't be similar enough. I'm okay with this. Cas9 is usually very efficient. I guess it sort of depends on where this mismatch is. If we, it actually shows us. So if I hover over it, it shows at the top. I can't move my mouse or it will go away. The sequence that it's suggesting, and at the bottom it's showing the sequence for the other target, potential target. And you can see there's 2 little stars. Those are the 2 little differences. Thankfully, the underlined part is super, super important for the Cas9 protein to sit and then cut. There's a mismatch, meaning it probably won't. So I think we're good. Also because we're not doing this for real, so it doesn't really matter. All right. So, why? What happened? I'm missing my my scroll bar to the left and right. So I'm just going to copy this. So I'm going to copy this, go back to our gene, Control F, Control V, and find it. All right. So that's kind of far from where I wanted. That might not be what I want. So why? There we go. Okay. So 93 reverse. 93 reverse. Okay, so it's right there. I wanted something more closer to 50. Ooh, okay. So we might go with 60 right here. Actually, we probably are. So there's a couple more off targets, but I think it will be fine. It will definitely be fine, actually. Oh yeah, that's fine. We're going to go with this one. We're going to go with that one because if I go back to snap gene, Control F, Control V, next, right there. So we are right over the deletion. So I'm going to mark that. So this is our guide RNA. Change the color. Okay. So that's our sequence. So the reason why I wanted it to be as close as possible to this deletion is because it will cut. So it will cut like right here, right? Actually more like right somewhere over here. After it cuts, what happens is that the cell will resect, which means it chews back the DNA on both sides a little bit. And that resection can cause a deletion, which is partially what we want, but we're going to give it a little bit of a heads up. So that is component 1 down. Component 2 is the protein, which we will just add in. There's a third component that we want to have just to make everything work just a little bit better, which is this right here. It's what is going to be called our template DNA. So when you cut DNA, the way that, not just CRISPR, but other genetic engineering techniques work, when you're talking about cutting the DNA is that it looks around. The cell looks around after its DNA has been cut and tries to find the homologous chromosome. The homologous chromosome is the other chromosome in the cell because it's really unlikely that both are cut at the same time. Because remember, you get one set of chromosomes from sperm, one from eggs, or the egg. So you cut one, it looks around for the other one because the cell knows that the other one probably has the correct sequence. So it will just copy it. That's what it's looking for, something to copy. Thing is, we can insert our own template to take the place of the other chromosome. It won't happen every time, but it usually happens enough that we can actually get the mutant that we want. Typically, what this is a bit of DNA that is homologous or identical to the area that we're looking for, plus and minus 500 bases around the mutation. Thankfully for us, our mutation starts. I gotta find the junction. All right, so that is our 5', that's the direction junction. And then this is our 3' junction. All right, so these are the two sides of our mutation, right? Because we have a deletion smack dab in the center. And so we want about 500 bases, give or take, on both sides. This one's about 500. And this one is at about 500. Perfect. Literally couldn't be easier. So now we have this, so we need to stick this into a plasmid backbone. You know what? This gave us a plasmid. What is this? I don't know what this is. Multifunctional enzyme. We don't need these things. It gave us it gave us some things. We don't actually really need them. But what we'll end up doing is we might as well just take the whole thing. Yeah, because you know what? While we're at it, we might as well engineer this protein to also have GFP in it. So this protein right here, so this file just opened with SnapGene, apparently just comes with it. This right here is GFP, or green fluorescent protein. We might as well maybe engineer some GFP in so we can actually find cells that have this mutation. Because why not? It's a non-functional protein. So if it's made, we'll have glowing cells. So this should be straightforward. We will do what's it called? We'll do Gibson. So we'll insert a fragment. So the bit that we want to delete is not that. Whoops. No. No. It doesn't want me to delete things. Select inverse. There we go. Well. Come on. I'm just trying to select just the right base. I'll go to the sequence. Okay. Invert selection. All right. So we're going to replace all this stuff. Oh, you can't see it. Sorry. I was clicking around this trying to decide what to delete. So we're going to get rid of all of this stuff. Get rid of this. linearized by PCR. Then our fragment is going to be this. It's just going to be everything. Product, make some primers, and that's how we make it. It's out of frame. Why are you out of frame? That means it wouldn't actually make the correct thing. We're off by 1. Okay, go back. Wait, do we need one more, or do we have too many? No, that's correct. Wait, what? Premature stuff. Yeah, it just... Oh, duh, the premature stop codon. You're right. You know what? You're right. But, maybe the other fragment could get picked up. Like, maybe another start would pick it up and make it like a little tiny protein that's no one's ever detected. I don't know. No one's tested this. So this is testing a hypothesis too. Because you're right. I forgot about the premature start codon. Or stop codon. All right. So we just need to get rid of one base. So let's go back here. Invert that selection. I'm only going to try this once more because it's not for real, so I don't need to make it perfect. Still got that fragment. Ah, there we go. Okay, so now it's perfect. So what is this going to be? So this is going to be our CRISPR donor. Nice. And there we go. That's it. So that was all we need for CRISPR. So this seems silly, but like what we just did was design the only 2 things necessary to modify the human genome if you wanted. Now it's not perfect. Like this vector, probably not the best thing to use. I just kind of used what literally opened up with the program. But what will happen is that this guide RNA, which granted has to go into another plasmid all by itself, I just don't want to deal with that. That guide RNA will cut right there. So it will cut right there. And then, so if you pretend this is the chromosome, the cell will look around and look to see what it can use to fix it. And well, at some frequency, it might see this and think, oh, that's pretty good. So it will copy the left and right hand sides here. Maybe it will pick up this GFP, maybe it won't. But you can imagine, this will be integrated back into the DNA. Maybe this, maybe the whole thing. It's hard to tell. Whoops. But the key is that once it inserts this bit, now we've introduced this premature stop codon right here. Or this right here. Oh, there's a premature stop even closer than I thought. So once you introduce this deletion, this premature stop shows up, kills off the protein. And that's it. That's all you need. All right, so that was a very long-winded example, but that is literally how you do CRISPR gene editing. It's a lot of effectively Google searching. It's not hard to plan at a basic level. It's hard to do, but just conceptually figuring out the pieces that you need, that was it. I mean, anyone can do this. If you're interested in giving it, a try, if you look up CRISPR, a plasmid editor, a free version of the software that I was using that I just honestly didn't want to download is called Ape, a plasmid editor. It's great. It does some of the same things that I was showing. just not as well. It's not as pretty. But that's it. That's all you need. Okay. I don't want to get rid of that quite yet. CRISPR. So, where was that? If I can find the paper that Brian sent us. I might have to reopen it. I think I closed it, because it conveniently had some positives and negatives about this. I think I closed it. There we are. All right. So why would you maybe want to do this? Why maybe would you not want to do this? So advantage, HIV protection and prevention in European individuals. So that's another good key. So most of the genetic information that we have, have in a bank, in a digital bank somewhere, and B, have studied is of European descent. We do not have good coverage of literally any other ethnicity besides Western, but probably also Northern European. So really, anytime we make claims about certain things, like the CCR5 delta 32 mutation, the problem is there's a problem, which is the background. So everyone has mutations all over their DNA. On average, doesn't really amount to much, but every once in a while there's synergy between them. And the problem is that maybe only CCR5 delta 32 works well in someone who has a set of mutations that are also highly not enriched, are very common in people of Northern European descent. But in the absence of those, maybe it doesn't do anything, or even worse, maybe it's bad without those other mutations. We just don't know, just because most studies that have been done historically within the past 20, 30 years with looking at population genetics of humans is done using Europeans or people of European descent. Apparently, there's also protection against other viruses like smallpox and flavivirus. I did see this, and I don't know what it means. Not what it means. Why this happens. Don't really want to look into it, to be honest. Is that people have faster recovery after traumatic brain injuries. So post-trauma and post-strokes, which is interesting. However, there are some disadvantages to this, and one of them, a big one that isn't mentioned here, so reduce protection against influenza, which, you might say, like, well, if I can't get HIV or smallpox or flavivirus, maybe it's worth it. Well, influenza, I'm pretty sure worldwide has killed more people than HIV has. Maybe not year to year, and certainly not in particular decades, but I think just overall. So I don't think you want to mess with influenza. You also get an earlier manifestation of West Nile virus infection, and then apparently there's an ambiguous effect on osteoclasts, so a type of cell that's found in your bones. The thing that isn't listed here, or at least I don't think it is. No. Maybe it's encompassed in this bottom part. I just don't know enough about it. The thing that isn't shown here is the increased incidence of aneurysm. So people with this mutation are at much higher risk for a certain type of aneurysm than people who don't have it. So that's obviously a huge concern. I just don't know how much of it affects the aneurysm chance. And also it gets down to an ethics thing. So this one patient who they where is it? Who they cured of leukemia and may or may not have cured of HIV, or at least reduced the effect of the HIV virus. Granted, that's not changing all of their cells, so maybe they are protected. Maybe the risks of that aneurysm far outweigh the downsides of dying of leukemia. Let's see. Yes, there's this stuff about Chagas cardiomyopathy. I'm not sure what that stuff is, but apparently point mutations in CCR5 have issues with chronic heart disease. So not good, not good. All right. It's still a really cool potential application of genetic engineering. Sorry, I keep clicking around because I keep losing this paper. Here it is. And I don't know how personally I feel. Okay, this I feel like was okay in a way, because what they were doing was they mutated the genetics of foreign cells that were going to be part of a life-saving procedure for this patient. This person can go off and have kids and will not pass that mutation on to their children. So it's not in the population, right? It's not going to continue on past this one patient. So in that way, as like a therapeutic, it seems like a really, really good possibility. Let's see. I am curious about the HIV thing, though. Do they have any data? Hemoglobin, CCR5 modified cells remain stable. Oh, here we go. Well, that's maybe confounded from the cancer, the leukemia. I'm just wondering, HIV, how they measured HIV. And this isn't the only one. I know more recently, so this is a targeted mutation, right? I think more recently some reports came out about someone who got a bone marrow transplant from someone with this natural mutation, or this mutation naturally, who was then cured or of their HIV infection, or just went down to undetectable levels of HIV in their blood system. Off targets, blah, blah, blah. Oh, here we go. Measurements of viral load. Clinical outcomes during and after the interruption of antiretroviral therapy. So they did have to stop taking antiretrovirals for the procedure, and probably for the leukemia treatment more broadly. Okay, that's... So let's see. Viral load, and then CCR5 modification level. So they did interrupt the antiretrovirals, but it seems like after this day 294 or 249, they went back on and went down. So did it work? Is the question. Their quick rebound after interruption of the viral load coincided with the appearance. All right, so it seems like this person actually still has HIV, but this seems like more of a proof of concept that they could give this person CCR5 ablated cells that maintain a certain level. So that's what this graph is here below. It's low, but it's still there. Oh, if genetic engineering as a cure for HIV, etc., will ever take off, I'm sure they'll be looking for the optimal genes to edit with as little side effects as possible. Oh, absolutely. But the issue is it's not as easy as just looking for specific genes, because a lot goes into genes. So if we look back here, right, so this is a particularly a pretty easy location in the genome. So I know it is confusing looking, but basically the gene starts at this green bar, ends at that red one. Pretty straightforward. You cause this one mutation, and then this gene gets shorter, and it doesn't work. It's pretty easy to understand, pretty easy to study. But not every single gene is as simple as this, and more than that, even if we stop thinking about individual genes, there's the rest of the genome to worry about. So, genes interact with other genes. That's just how it works, or more the gene products of other genes. So changing one, the cell is pretty robust, but changing one is not likely to only change one thing. It's going to have sort of ricochet effects that we won't ever know without proper study and actually doing some of the mutations in question, whether it's in like mice or human cell culture or in humans. But otherwise, absolutely, like you're right. As this becomes more common, we are going to look for genes that we can modify with as few consequences as possible. I think the best examples are probably, genes that can be mutated in people to cause really severe disease, and we know how to mutate them sort of back to a state that is not disease forming. So a good example is Huntington. I think that's what it's called. So Huntington, where are you? Huntington is the causative agent of Huntington's disease. And I don't even know if this reference genome is going to show it. Where'd you go? Because also I don't quite know where it is. Oh, am I looking the wrong direction? I got to find the right part of this gene. So Huntington's disease is progressive, it's familial, it's really awful. I think, what is it? I think you have to be... No. People who have 2 copies, one from both parents, are dead. I don't think they ever survive. People with 1 copy have Huntington's disease, which is a progressive degenerative disease. Where is that mutation? I probably have better luck just looking it up. So there's this, basically what happens is this gene gets longer and longer and longer through a process of like slippage, which is really strange how it happens, but it's interesting nonetheless. Huntington. And the thing is, like, we know that this happens, and we know that if we are able to disrupt it, then a person will be completely fine. Oh, here we go. It's in the middle of the gene. Yeah, so this is a good picture. It's not the sequence, but it's close enough. So you have the protein, and in the center here are these CAGs, CAG, a bunch of them, and that's what you need. Unfortunately, in some people, these expand, so you suddenly get hundreds and hundreds of these things rather than a couple dozen or even a dozen. and this causes Huntington's disease. So using something like CRISPR, it's very straightforward how you would basically fix this, right? You'd attempt to cut as much of this as you can out of it and sort of repair it in such a way that it looks much more normal. And potentially you can save not just, well, honestly, probably not the person who's affected, because I think it affects brain tissue, which is not something you want to be CRISPRing. But you could certainly do something to perhaps save, like modify their sperm or their eggs in order to save their children from having the disease, stopping them from passing on the disease. Okay, I'm looking for attractive cues. All right. But I did want to talk about this in like the context, in the context of sci-fi, though, because certainly this is not the realm of just, real life. People have been talking about gene editing for a little while, even before like the technique of CRISPR came about. So we've been actually gene editing probably since, what is it, the 40s or 30s or something like that. What we used to do is you used to have a teacher in high school who had a parent with Huntington's. Yeah, it is really sad. From what I understand, the sort of decline of people with Huntington's disease is just awful to watch. They just deteriorate mentally, and it's really awful. So, in terms of like targets for gene editing, I think Huntington's disease has to be at the top, because if it can't save people who are carriers for it, not carriers, they have it, because there are no carriers. If not to save people who have it, it will save the lives of their children and their children's children. Anyways, so gene editing actually kind of started a long time ago with nuclear gardens, I think they were called. Atomic gardens, excuse me. Basically, and I feel like I mentioned these before at some point. Basically, and this is one, I think, in Minnesota. They sort of still exist. What they do would plop down in the center of this something that irradiated or gave off radiation, ionizing radiation. Then they'd plant crops in a circle around it, because ionizing radiation, comes out from all directions. Although by about this point, you're not really receiving a huge amount anymore, so that's why it's relatively safe. And then bombard plants with ionizing radiation with the intention of creating mutations, so that you can then take those plants that have mutants and look for positive traits. And we actually still use grapefruit and a type of mint that were created using this method. So David says, what about early screening and proactive gene editing? Can't that save people? And then Miss Frizzle says, yes, that was her thing. She was talking about how she was going to have screening done, even though she was in her early 20s, since she didn't want to put the future child through what she was experiencing, but also talked about the conflicts in wanting the information you can get from genetic testing. Yeah, so in my human genetics course in college, we had a speaker come in who was the child of someone with Huntington's disease, meaning that they had a 50-50 shot of also having it. This person chose not to find out if they were a carrier, which is interesting, but it's her life, her choice. I do feel bad. Like, that's like basically a lose-lose situation. But yeah, so, but to David's point, and yeah, it is understandable, like so understandable. So early screening, yes. So we can do screening, but as Miss Frizzle said, even now with the advent of like CRISPR technology and various forms of proactive gene editing that might take place, it hasn't yet. So all screening prior to some future date when we are proactively gene editing people, has really only been done to tell you whether or not you should prepare for the worst, or if you, like as Ms. Frizzle said, need to find out whether or not you are going to put a child at risk of losing their parent and or having this debilitating disease themselves. So we can screen for it, but the proactive gene editing, that's tough. And it would have to be proactive pretty early. So one of the issues is, so like, let's take that example with the bone marrow transplant. So right, the bone marrow transplant you take bone marrow out, you do some CRISPR and basically make this mutation, right? So the problem when they tried that, right, was that only about, what they say, 5 to 8% of the cells had the mutation. So that was when they had a specific tissue in the lab in their hands, and they were only able to get like 5% out. With some diseases like Huntington's disease, where it's a malformed, misfunctioning protein that is causing issues, you really have to get the majority, or at least a huge proportion of cells to change. And at the moment, we don't have a way to go into a living human being, especially an adult, and mutate every single cell. The best time to have done that was when they were a single-celled zygote or a sperm and or egg, right? So, let's say we had a sperm donor and an egg donor. The egg donor has Huntington's disease. You have two options. Sort through the eggs to figure out which ones have the Huntington's disease allele or which ones that don't. Let's say for some reason you don't want to do that. You could go into the egg cells and maybe use CRISPR to change, the gene. And then you fertilize and then you're off from there. That's really the only guaranteed way. But then it gets into sort of a problem, which is kind of the, where'd you go? The Gattaca, I don't know if it's problem, but as soon as you start getting to this level of changing enough cells to save a person's life from something like Huntington's, you also get into this part where it's like, okay, all of a sudden I'm probably mutating their gametes. So you're taking away the choice that they have, although that seems a little funny because you're saving their future children from a life of disease. Still, you're sort of taking away a choice. It's kind of philosophically, I understand the arguments sort of against that, even if I don't agree with it, but that's an argument. But also you're just getting to the fact that you are creating a evolutionary path that didn't exist before, right? So if I had a disease and my gametes were CRISPR'd, but not me, then all of my descendants for all of time will have the differed genetic mutation. Maybe not all of them, because, genetics and all that. But still. So it's this I don't really know. It's sort of tough in that way. So I hope that sort of makes sense. Ooh, I didn't even see this. So, this article is actually pretty good. I was talking about atomic gardens. We don't have to talk about atomic gardens, although they're really cool. Because I swear, I did want to talk about sci-fi, but wow, it's almost 10. I've sort of mentioned it, but not too much. I had too much fun looking at my own genetics and then doing some CRISPRing. So, I guess let's just scroll through this, though. I do want to see what this article has to say. So again, because Gattaca kind of encapsulates a lot of the sort of problems or issues that come up. Because this was all about making designer babies. And I think here they describe them as a eugenicist society, so a society that shapes itself based on the genetics of its people. The sort of problem with this for all of history, and that will still be a problem in the future, is that genes do not necessarily equate to outcomes, not in the way that matters for society as a whole. David, you say, considering that by purely natural means of conception, there isn't a lot of choice involved either. You don't see the problem, at least if you limit any genetic engineering to curing diseases. Yeah, so it's kind of tough. I think I agree, because I'm very much in the camp of, if you can reduce harm and suffering as much as possible, that is only a good thing. But I recognize that there is an argument against taking away the choice and agency of future peoples. That seems silly, right? Especially when you're talking about a disease that will, by your 30s or 40s manifest as a cognitive decline requiring full care before you hit like age 50 a lot of the time. That seems silly, but that's because Huntington's is such a clear choice. If you start sort of moving the needle back, well then what do you say about like glasses, right? Once we get rid of all the bad diseases, what if we start talking about nearsightedness, farsightedness, that sort of thing, and then it's literally the slippery slope. I think there's some very clear positive outcomes for some very distinct diseases, but then after that it gets a little wishy-washy. For example, the CCR5 thing, right? So let's just say that we decided that, increased influenza, maybe this weird aneurysm thing, those are fine. Let's rid ourselves of HIV. Okay, we do that, but then, a generation later, we find out, oh, wait, actually, there was a reason why CCR5 evolved and was maintained. We actually really need it for XYZ, right? We just do not know enough about genetics and biology to have the complete picture that we need to make truly informed choices. I don't know. It's hard. We can make super, super smart, educated, reasonable, not guesses, hypotheses and inferences, but we're just not there genetically quite yet. And related to that slippery slope, right? So Gattaca is in a world where we've gone past that slippery slope. We've cured a bunch of diseases that we can cure, but now we're into the territory of designer babies, something that is like on the media's mind and has been basically since this came out. Yeah, Mr. Rizzle. No human genome reduction project. As much as I kind of, hate all this wasted space. Ugh, look at that. Oh, look at this gene. Actually, look at Huntington. You don't need all those exons. Much as I hate it, probably shouldn't go poking around in there too much. Oh, thank you. I think it has been a very good conversation, too. I'm glad people are interested. You bet George Church wants to. Yeah, no, I bet George Church wants to make like a single chromosome, all exon / CDS human something woolly mammoth. He probably wants to replace all human genes with woolly mammoth genes or something like that. I don't know. Although, no joke, I would work for George Church in a heartbeat. He has the money. He has the money. Doesn't seem to care what he does with it. You say you get it, but it seems like with any other advancement in medicine, a lot can only be learned by practice, a lot of potential for abuse. I just doubt finding out where to draw the line would be impossible. Oh, I see. So you're saying you think there is a way we can find where the line is. Yeah, no. No, David, I am totally agreeing with you on all fronts. I do think we should just kind of go for it on some things. That doesn't mean I can't have concerns, though. I think we should do things, but then keep an eye on like, hey, is this taking away our future children's agency? Is this maybe are we getting in too deep too fast? Are we doing a Gattaca? If we're doing a Gattaca, we got to pull back. Which, by the way, interestingly, so this line right here mentions something which I feel like is kind of lost in discussions about Gattaca. So Gattaca, absolutely. It's about like there's a scene where parents come in and they're talking with someone and they're like, okay, so you want your child to be tall? Yeah, we'll just make them tall. You want them to be athletic? Yeah, we'll make them athletic. All this stuff. But there's also this notion of that's like in addition to this other thing, which is screening. So pre-screening children for diseases and traits and not pre-screening them. So that itself is kind of an interesting subject, like, because we do that now for disease, and especially certain types of birth defects that will result in loss of pregnancy and possibly harm or potential death of the mother. So those are like really required. But I think even without the genetic engineering, if we start being like, oh, well, they have the tall gene or they have the muscle gene, that can get a little squishy too. You got the NIPT. Sorry, I'm laughing because I saw your misspelling before I even got to it. You got the NIPT when you were pregnant. Actually, can you speak to that? Is that I don't want to say required, but was that just kind of like assumed that you were going to get that? Like you went into, OBGYN and they were like, okay, you're already signed up, you're going to get this, you're going to get that. Or was it an opt-in or opt-out sort of thing? I'm just kind of curious. Yeah, pre-screening is, I think, really important. But again, let's say we threw in, what is that muscle gene? Strong cow muscle gene. How strong is Goku? Yeah, because this is what that CRISPR, that biohacker guy was trying to do. Like, what if? Okay, here's a business idea. Let's all go start a company. We'll do the normal, prenatal screening and all that. But our thing is we'll also screen for this myostatin, mutation in the myostatin gene. So we can tell potential parents, you're going to have a super muscular bodybuilder baby. That will be our niche. Not required, heavily encouraged, and covered by most insurances. It was very opt-in, but you've had previous pregnancy complications, so they really encouraged it. Okay, that's good to know that they encouraged. I mean, with someone like anyone who, I don't see a reason nowadays why you shouldn't. First-time mothers, older mothers, people with complications, people with just the means to get it. It just seems like it's something you should get. But yeah, so anyways, like we could start a company that throws in the myostatin mutation gene thing. And suddenly I feel like we've crossed a line into like pre-screening for things that, maybe you could say is important or bill or promote, but you do not affect at all and isn't actually important. And suddenly we get into this territory that I think Gattaca touches on but doesn't focus on, which is very interesting. The idea that like maybe you could know something about your child before they were born, which is, I think, if people agree with me, genetically untrue. I mean, there's simple things like hair color that of course you can tell beforehand. But that's like not very impressive. They basically require it for twins based on your sister-in-law's experience. Interesting. And you can get the proper care team at birth if you do genetic testing. Yeah, that makes sense. If you find out that you're going to have possible complications, that's good to have. Yeah, I'm not saying anything against pre-screening. What I want to point out is the fact that we're already like the first step towards part of the Gattaca thing, and all it will take is, an unscrupulous group of people who want to monetize pre-screening. I just remembered something, and I was trying to download it. Why? Again, with my DNA testing thing, the top results of like, hey, we're most confident in this was the fact that I have a Y chromosome, red hair, and brown eyes. Those were the most confident things. Oh, and light-colored skin. I don't need to show that, though. They thought one of the twins had a high chance of Turner syndrome. And it was just so like, hey, you can go ahead and start forming the doctors you want to work with early, which can be very helpful. Yeah, that is super helpful. That's good. Yeah, I haven't had the experience of needing to be in consult with neonatal testing. But that's interesting. I've always kind of wondered, yeah, like what are all the things that they look for? All right. So let's skip down to like, this is the science realistic? Right. This was the screening part. So they do some IVF, real technology, right? And then they had multiple embryos, and they selected the most competent candidate, which I swear I've heard has been done. Someone should correct me on that, and I could probably do some Googling really, really quick to figure out. Do you select which embryo you want? Oh, IVF and gender selection. Oh, I did not know this. Wait, I did not know this. You do this all the time for cows? Yes, I do know that. But, and sexing sperm. Yeah. No, I know you can do that. Oh, I do not need my modern guide to parenthood. Thank you. Decline cookies. Decline. I can't decline. There we go. I'm actually shocked now to learn that we can do that, or we have been doing it with people. Simply put, family balancing means that if the intended parents always wanted a girl but only had sons, they can select gender during IVF to ensure a balanced family. I feel like they should have put this next one on. At risk to pass on sex-based genetically transmitted disease. I feel not good about this. Dairy farmers joke about it. To have boys to work. I roll. Do you how many like cow sexing jokes do you know? That reminds me of, I forget how she put it. So I have a cousin who a couple of years ago was like young. Oh, this would have saved so many of Henry VIII's wives' heads. Yeah. It was Henry VIII's fault. He didn't get he probably wanted a son, right? I just assume. Sperm provide the sex chromosome required to determine biological sex. Barring syndromes where you get multiple sex chromosomes from one gamete. So my young cousin, she is like, at the time was like 3 or something. Her parents are both farmers, and she had been around the farm, and she had been around when they came around to, I don't know what the term is, Miss Frizzle, if you know the term, but basically when you take the straw of bull semen and impregnate, oh, that's it, impregnate, duh, and impregnate the cow, right? So she was aware of this. She knew that like a man came with these like straw shaped things and would impregnate cows, and once you impregnate the cow, a couple months later it would have a calf. Like she knew this. She was like 3 or 4. Well, then her parents, they get pregnant, right? And she finds out she's going to have a little brother. And apparently, as a 3 or 4-year-old, artificial insemination, but I think they just called impregnating. Oh, no, she did say inseminating. Anyways, I'm telling the story awfully. But basically, she came up to her mom one day after her mom was visibly pregnant, and apparently she had thought about it for a while. And she asked her mom, "So, who inseminated you?" Which I was absolutely love, and that's going to be something we're going to tell her constantly when she reaches her teenage years. Anyways, which is, first of all, fantastic. Like that like child logic of simply understanding and accepting the series of events required for a process to happen, such as pregnancy. Also, a very big word for a preschooler, inseminate. I think it was funny to them until she started telling her like preschool classmates that someone impregnated or inseminated her mom. Then you have the vet come to do palpitations to check for pregnancy. No one can afford that fancy stuff. Well, I think at least my cousin's wife, who was pregnant, I think they've got the fancy ultrasound. But yeah, no, I don't think they did not a super fancy dairy cow farm. So I'm sure they, except for the straws thing, the insemination thing, they have a pretty old-fashioned way of going about it to get more cows, or calves, I mean. Gender selection. I'm actually, okay, interesting. Okay, this is just about gender selection. Okay, anyways, competent candidate. They even get it. They even go right here. They mentioned it. I just got distracted. So they also mentioned, checking the DNA of the embryos and choosing not to transfer ones that have high genetic risk for some diseases. So I did know that. Oh, interesting. So we can now screen out Huntington's disease. I bet this is really expensive, though, because IVF, first of all, is super expensive. So this is definitely going to see already, though. So Gattaca is all about the formation of 2 classes based on access to genetic services. That definitely happens now. I mean, the couple who goes in to get IVF and then to select an embryo that does not carry the Huntington's allele probably is fairly well off, and that helps like further stratify class barriers. So that's problem. And they say, but not more common issues like heart disease, asthma, and cancer susceptibility. So they say that, but I really don't know like what's keeping us from trying. Like again, I could start a company where I basically just have SNPedia pulled up on the side, do my own, not do my own, I would farm it out. I'd farm out my own genetic testing service for pre-implantation embryos. I just kind of scroll through these and just tell them like, oh yeah, that one's got a higher risk for this and that. And suddenly, with this new company that I just dreamed up, we're making decisions based on genetics, trying to screen out these more common issues. Oh yeah, it's very expensive. So this reminds me, or you, of this book series you read called Uglies. It may have been inspired by the movie. Is it called Uglies or The Uglies? Ooh, 2005, 2007. I think that's definitely after. Wouldn't you have to destroy the embryo to get DNA to sequence, or how would that work? So I think the embryo is actually like a blastocyst. So you can. Yeah, so I think um, the embryo at this point is actually a blastocyst. So it is this right here. So at this stage of embryogenesis, this inner portion, this inner mass, is what actually becomes the embryo. This outer mass, although it is made of embryonic tissue, does not become the body of the embryo. It's the trophoblast, which becomes, I believe, becomes the placenta and umbilical cord, and maybe like the amniotic sac and all that sort of stuff. I don't know if this was like clear to people, but something that shocked me way too late in life is that the umbilical cord and placenta are part of the baby, just not the part that we keep. So it's not a part of the not a part of the mother. It's born of the embryonic tissue. Uh, yeah, you weren't sure how far they could culture them. It's around this stage, maybe before. They can take a cell or two, basically. You remember the uglies. Oh, your body has problems with this process, and you both enjoyed it. Yeah, the uglies. I see it's a trilogy. I feel like I remember something about the pretties. You're so upset that you did not get to see yours. Like, you mean your pre-implantation embryos? Did you ask? When I donated bone marrow, I asked if I could have a sample of my stem cells, and they told me no. They did let me take a picture with them. Oh, you want to see your placenta? I see. They wouldn't let you, or maybe you were too. They just. Oh, that sucks. That seems like they should have been able to see you. They had to get it to pathology, so they just took it away. Fair. Someone could have. Someone could have taken a picture or something for you. And no, don't listen to Megan. Don't make it into pills, please. It's consuming part of your child, the unwanted part of your child. The Tom Cruise approach. Oh no. Yeah. Uglies. I will have to look into this. Tally Youngblood. That sounds like a YA novel. Is this YA? Yep. Yep. That's a YA novel protagonist if I've ever heard one. Anyways. Oh, yes. I mean, it kind of sounds like it. All right, so I do have to get going soon. So I know we didn't talk that much about sci-fi, which I'm a little disappointed by. So maybe I'll touch on this later, but that's okay. We had fun. What I do want to say though is like this person who has, I'm glad they talked to someone smart. I don't think the premise of Gattaca is close at all, although I do think that this Greeley person missed the whole screening process thing, because you could totally implement screening even if it genetically maybe didn't amount to much. That's still sort of the pretense of part of Gattaca, is that despite the genetic superiority of some of the characters, the people who are not as genetically superior are equal or better in other ways, which is because genetics is hard. Yeah, genetics is not that powerful. But the premise of the movie is not about the like actual genetics. It's about the society built around the idea that genetics are that powerful. Most diseases and definitely traits occur because DNA variations because of DNA variations and environmental influences. These cannot be programmed into DNA. Well, some things can be. I know this person is just speaking. I saw they were a professor of something or other from somewhere. This person is correct, but also this is the thing. Sometimes it can be programmed into DNA. Where is it? I don't think I have it still. Somewhere I have a picture. So I have red hair. This right here, we can go in and find the red hair gene. Like it's pretty straightforward. So let's go find it. It's RS1805009. I don't know if it, nope. Sorry, that was probably pretty loud. 007. Nope. 008. I know I have it somewhere. There it is. Actually, that's not quite right. I have some mutation somewhere that I can like very clearly go and check. It's like, oh, that's why I have red hair. Anyways, so some things in DNA are determinative. I don't think I guess who knows? What if I ate a different diet? Maybe my hair wouldn't be red. I don't really know. Gattaca's use of genetic screening to precisely predict a child's tendency towards mental health issues like schizophrenia or ADHD is also implausible for now. So thankfully, intelligence, IQ, performance, and cognitive tendency toward depression are not one gene. But, an orchestra of genes. That's a good way to put it. But, I mean, I think the last thing I want to stress is just like, no, it's not. But also, oh, look at this awful picture. But also, like, if you convinced enough people that it was, you could set up a company to, like, pretend like these things are important. And not do a Gattaca for real, but simulate a Gattaca where a good proportion of people would, like, talk to their friends about how their child is genetically superior or like, has the best traits or something like that. This is a stupid picture. Very stupid. Is the DNA even going... It's not even spiraling correctly. All right. Okay. So, we got through exactly 1% of the science fiction that I wanted to get to, but we talked about a lot of real science, so that's okay. And we got to look at some of my own genes, which is fun. The future will be in AI improvements. Imagine if you have a system that goes over any given genetic sequence and translates it for you in no time. I mean, translates in terms of like function, maybe someday. Yeah, that'd be cool. We are gaining more and more information in the lab now for what specific genes do and the structures like this that give them their function, right? The structure-function connection. And maybe with enough data and a good enough, basically enough data as a training set and a good enough AI, maybe you're right. Maybe we can start predicting things like that. Although, cellular function and traits are emergent properties of these various proteins bashing into each other inside the cell. So even if you had a AI program that could predict the structure and maybe the function of a protein, if you don't know that this other protein is over here, maybe you don't actually truly know what it does. Not disagreeing with you, but I think we're a little ways off from that. But it's going to happen. I think AI is definitely the answer for stuff like that. All right. I think that has to be it for tonight because I have to go to bed. But thank you all for coming. I think Sunday what I'm going to... I think Sunday what I'm going to do, I don't know if it's worth it. I want to make that tardigrade maze. So if you missed it, what I, oh, thank you for the reminder about a raid. What I want to do on Sunday is design and print a little tiny maze for tardigrades. Because I got all these tardigrades and we got nothing to do with them, except for watch them, which is fun. But if I can print a small enough maze, maybe we can make them like journey through it. This is all assuming that my printer can print something small enough. We'll see. Oh, Embis is on? Oh, great. We'll go raid Embis. Thank you for checking. All right. So stick around for the raid. Embis, why don't you show up on mine? Yeah, stick around for the raid. Otherwise, thank you for coming, and I'll see you Sunday. Bye.